through sexual contact. HPV particles then access the basal cells through microwounds in the cervical epithelium. The HPV virus penetrates cells by binding and interacting with the surface of the host cell. The virus replicates from episomal DNA to form new viral particles in the upper layers of the epithelium. This benign productive process leads to the formation of cervical lesions and to the release of new HPV viruses that can then initiate a new infection. Most HPV infections are benign, but some may progress to cervical cancer. The starting point is identical to a benign infection. However, risk of malignant progression increases in cases of persistent infection with high-risk HPVs. Due to factors not yet clearly identified, the DNA genome of the virus integrates into the chromosome of the infected host cell. This results in DNA rearrangement and overexpression of two viral genes, E6 and E7. This abnormal upregulation drives persistent production of E6 and E7 proteins in the host cell cytoplasm. Overexpression of E6 and E7 induces the inhibition of p53 and retinoblastoma proteins, two key proteins controlling normal cell division. Disruption of these functions leads to disturbance of the cell cycle, uncontrolled cell proliferation, and subsequent progression to cervical cancer.